So, Carmen, won't you take us through what the targeted hyperthermia therapy here is?
So targeted hyperthermia, what this essentially is we're taking the gold nanoparticles, we're injecting them directly into the tumors, and in doing so, this is a preclinical mouse model, of course, the standard of care for all immunotherapy. We grow a tumor on the animal, then we fill the tumor with gold nanoparticles. We don't give it systemically at this point because we just want to concentrate the gold in the tumor and not be concerned about distribution throughout the animal. That's a safety feature, and it actually helps us to understand what we're seeing. Then when the tumor is infiltrated with the gold, we pass near infrared light, laser energy, through that tumor. The gold that's within the tumor absorbs the energy. The laser is about 860 nanometers. The gold particles are about 60 nanometers by 15. So this combination of things like a tuning fork, when you pass the energy through the gold nanoparticles, they vibrate. The vibrational energy creates heat. So it turns the light energy into heat. And the heat, and we monitor the heat so that the heat never goes above 48 degrees. That's like high fever heat. Now, extremely high fever heat, I'll give you that, but that is for cells of the body. Your cells can tolerate 45 degrees quite comfortably. Not you physically not comfortably, but the cells don't. No harm comes to them. But in cancer cells, they're not very well stressed, and so when we heat cancer cells, little proteins in the cancer cells called heat shock proteins are unreleased. And in releasing heat shock protein, we activate an immune response, an innate immune response. That is the immune response that responds to stress in general, and this being a stress. And so this is we're capitalizing, exploiting, if you will, a function of nature, which is to heat tumors specifically. And in the old days, in my practice, we would heat a whole body to do the same thing. Now we've got this very elegant, targetable hyperthermic therapy that we can put directly into the tumor. And in doing so, that causes the tumor cells to die. When the cells die, your innate system, the same system that heals a wound when you get cut or after you have surgery, that system comes in and causes a healing effect. It chews up the dead and dying cells. And in doing so, it constantly shows those dying things to the immune system so that your immune system knows if this is new, if it's foreign, if it's due to an infection. And if the immune system hasn't seen this protein, which is essentially what cells are made out of, then it will continue to send out fighters to eat that protein until it's gone, until it's cleared. And this is the innate immune response that we're hoping for. And thankfully, I can say we see it in every cancer that we've treated. So this laser energy is the elegance of it is, again, we put the gold into the tumor and then we heat the gold, passing the laser through it almost on a cyclical format, like a harmonic format, so that the temperature goes up and down between 42 and 48. So at about 45 degrees, we're comfortable that we're not killing other cells, and so we don't see any other cell death. The tumor cells die, all the cells around it are maintained. The heat is in for 5 minutes, it stopped, the stress goes away, and the natural process of cell death and immunogenic cell death, meaning that the cell that's dying is generating an immune response, carries on. And we've proven this in our experiments.
And that's a critical point you make there, that the gold nanorods, why they are the ideal and the superior nanoparticle for this application is that they have the most efficient conversion of non-thermal energy into heat. And so if we just turn on the laser and let it go, that heat will continue to rise up to the point where all cells are being damaged. And so this harmonic effect is keep it in the band between 44 and 48 in order to give it just what's needed to kill off the cancer while preserving the healthy cells.
Exactly. That's critical to the whole process.
Okay. So what's new? Let's get into this colorectal data. And Carmen, do you want to take us through?
This is again an industry preclinical model. This is a cold colorectal cancer, not the kind that we currently use immunotherapy for. And what we've done is we've taken this cancer, we've grown it in the flank of the host animal, and then you see this first line here. This is if you put the tumor cells into the flank and just nurture the most, the tumors will grow, and they'll grow pretty much unbridled. Slow, steady growth. You can see the way the curve goes. There's kind of an uptick. This is the old, think about it this way. If you have 2 pennies and then you multiply them, 2 becomes 4, becomes 8, becomes 16, and so on. So there's an exponential curve of growth. That's the natural curve of cancer growth. When we treat with PD-1 inhibitor, which is what that gray curve is, the PD-1 inhibitor, again, the checkpoint inhibitor that blocks that, if there's an immune reaction in that tumor, then the PD-1 inhibitor should enable that immune reaction and diminish the growth of the tumor. To what extent is depending on a whole bunch of factors. But what we see here is there's essentially no effect from treating these tumors with direct injection with a PD-1 inhibitor or systemic injection. We've done it both ways. Next. So this is, if you repeat the experiment, and we've done it a couple of times now, so we're seeing 2 cohorts, meaning we've got a group of mice, we do it to, we repeat the experiment with another group of mice. Twice we do this, and we see that if we first treat the tumor with targeted hyperthermic therapy, in other words, if we turn on that innate immune system so that the tumors are now being presented to the immune system, the protein in the tumors specifically, and the immune system now sees that as an immune target, then the checkpoint inhibitor, the PD-1 inhibitor, again, following the logic that we've talked about, will be now effective in an area where it wasn't effective before. And that's exactly what we see. Those lines are essentially flattened. So when you look at these, when you look at the tumor volume, you have an initial growth, of course. You have to wait until the tumor has volume to inject it. And then when you have an injection point, no more growth. So we arrest it. Now, what is some of that, when you look under the microscope, some of that volume is actually structure to the tumor, the supportive scaffold, and tumor cells within it are dead. But as an indicator, what we're seeing is we've arrested growth. Those are essentially flat lines. You see the blue line where things, now, why do the lines stop? That's a function of the experiment. When we write the protocols to the animal ethics people, we have a finite amount of time we're allowed to keep those animals alive for humane reasons, obviously. And so we're forced to terminate the animals and sacrifice the animals so that we prevent the suffering. So these are finite numbers. Essentially, our most recent protocol got us to 26 days. And if you the blue line again, we were really quite surprised. Not only did the tumors respond, the immune system completely cleared them. If you had an ulcer on your hand, envision the ulcer completely healing and the skin returning to normal. This skin was essentially normalized from the tumor. So that was above and beyond. Now we're excited about that, but that's, if you think about it, everything I'm telling you, the immune system should get you to that point eventually.
So now we wanted to break down, because these lines represent the average for the, it's the aggregated data for eight mice effectively in each one. What does it look like when we break them out? So we wanted to break that out for you a little bit and show, in this case, Carmen, this is just the cohort treated with immunotherapy.
Right. And so these lines, so we broke this down for you to see that these are mice that are these are tumors grown and then treated with PD-1 inhibitors. And what you see on every one of those curves is that the curves turn upward. There's lots of factors that go into tumor growth. When we show you an average, we're showing you a line that's an average of 8 lines. And so some tumors grow slower than others, some tumors grow faster than others. What we can tell you from these curves is that the treatment with the PD-1 inhibitor had no effect on the trajectory of those lines. They continue to go on in whatever growth rate that they had started with, largely because there's no effective immunity happening in those tumors. Now, we've also disassembled the ones that we treated with the targeted hyperthermia and the gold. And if you look at that slide, you see these lines. And so this is every mouse. That's 100% of animals treated have responded to this therapy. That in a laboratory setting is in and of itself astounding. So this is reassuring. From this data, all you can say right now is that targeted hyperthermia activates immunity, and without it, there is no effective immunity.
Yeah. And it's clear that in each case, the mice treated with THT did better than each instance of mice that had the best-in-class current standard of care immunotherapy. Right. That to us is compelling.
Yeah, and that's right. And that's exactly it. So again, this is essentially addressing current standard of care and showing that we can make a difference where it doesn't work.
Mhm.